Okay, welcome back everyone. So we continue the session. So my pleasure to have next speaker, Dr. Nusara Satproedprai. Dr. Nusara from Health System Research Institute. Nusara studied medical technology of Faculty of Allied Health Science at Chulalongkorn University in Bangkok, Thailand, and she received her PhD in microbiology and immunology with focus on molecular biology from Virginia Commonwealth University, where she developed her interest in gene expression analysis, gene signaling, and human genetics. After graduation, she joined the Department of Medical Science at the Ministry of Public Health, Thailand, where she continued her research in development of the novel method for infectious disease diagnosis using molecular techniques and study human genetic factors associated with susceptibility to infectious diseases. Nusara is also the founding members of the National Strategic Project on Population Genetics entitled Genomics Thailand, and the Committee of the Medical Technologies Council of Thailand. She joins Eastern Economic Corridor Office in 2021 to work on promoting investment in medical industry of Thailand. So it's very honored to have her to talk on the gene expression in TB diagnostic today. Please join me to welcome Dr. Nusara.
Thank you very much for a nice introduction, and also thank you the organizing committee of the HIV Symposium for a great opportunity for me to talk about my previous work with the Department of Medical Sciences at the Ministry of Public Health. So today my presentation is entitled just a little bit change for excitement, blood-based gene expression biomarkers, rethinking tuberculosis diagnostic. And I will address this topic based on 3 questions, which are, why do we need new tools for TB diagnosis? Second question is that how can we find a novel biomarkers for TB diagnosis? And are these new tools, even we find it, is it ready to be used in the clinic? So first of all, you may wonder why are we talking about tuberculosis in HIV Symposium, but again, tuberculosis is still a world's deadliest infectious diseases with 1.3 million annual deaths and 10.6 million new cases in 2024. And approximately 25% of the world population have latent tuberculosis or a latent state of infection, and creating a massive reservoir for a future disease. And to achieve the WHO 2033 strategy, a high accuracy triage test that can confidently rule out the disease or reduce unnecessary referrals may serve as one of the tools to help us achieve ending TB. And also to tie in with the HIV, TB is the leading comorbidity in people living with HIV. So what are the limitations of the current tools for diagnosing TB? So first of all, I'd like to show these slides because these are the conventional method that we use to diagnose TB. For example, sputum smear, MTB culture. These tools are, even though it's all like about a century old, but we still use it in some certain rural area and rural hospitals where we cannot find any other molecular methods to implement. So it's still for sputum smear, even though it's fast, but it has a very variety in terms of sensitivities and also for MTB culture, even though it's very specific, but again, the assay is very slow. It gets 2 to 8 weeks until the result can be returned. For newer methods of diagnosing TB, such as tuberculin skin test, or the most recent one is IGRA, or interferon gamma release assay. Even though it is very sensitive, but still has some troubles especially in a country like Thailand that we have a BCG vaccination since birth. So, and that reduce specificity for instance of TB. So, however, one can argue that now we have a very powerful tool such as MTB gene expert that can confirm whether the patient has TB together with drug-resistant information, but it can be challenging when it comes to tell the risk of TB reactivation or the development of a clinical stage of tuberculosis, or to differentiate it subclinical TB from other diseases. So in this, in my talk, I'm interested in using a novel method to tell whether a patient is developing or is having an active state of tuberculosis. So the blood biomarkers offers a non-invasive window into the host pathogen dynamics. And from these slides, and actually we can detect direct components on this side. We can even detect some pathogen-derived biomarkers such as RNA, DNA, or lipoarabinomanans, remnants of those actually in the blood of the patients. On the other sides, we can also detect a host-derived biomarkers such as RNA, cytokines, and metabolites from the bloods of the patient. And host markers can potentially be detecting the disease earlier and distinguish between the active states of tuberculosis against the latent states of the tuberculosis. And this area has been developing very fast in the last decades and show a promising data. So the blood gene expression analysis just provide us a global snapshot of a host response against infections. For example, even though the tuberculosis etiology site is in the lungs, but from lungs to blood circulation, white blood cells travels between the lung tissues and the blood cells, and those and they carry with it the transcriptional abundance of cytokines, chemokines, and other immune response against the diseases. And when we can measure that by simply collecting the blood. And these differences between the healthy states and the disease states of this blood transcription can help us understand the pathogenesis or identify therapeutic targets, or even ultimately improve the diagnosis. So for today, since this field of new biomarkers for tuberculosis has been developed so far since 2010, so I will cover these hallmarks studies before we go into my studies here in Thailand. So the field of blood-based gene expression profiling had advanced rapidly, and the first and foremost, the hallmark paper is from Berry Group in 2010, which is the first group to spark an idea of using these blood transcriptional biomarkers to identify different stages of TB infection. And this field of study has developed since then, and I will walk you through some of these key developments. So first of all, the hallmark paper from Berry Group, they study a transcriptional biomarker in the blood using a powerful tool called microarray that can explore the differential expression of 20,000 genes in a single run. And this paper identifies the gene that is expressed exclusively in the active TB. You can simply see here that these 394 genes has been turned off or turned down in the active states of tuberculosis, while it is differentially expressed in the normal people or the latently infected individuals. So by comparing these genes, we can identify a key targets that for use as a biomarker. And again, and this on the right-hand side shows the distinct pattern of gene expression between a different time post-treatment, for example. This is from the healthy controls, and these are from the active tuberculosis patients. And these are from the when they were diagnosed with the tuberculosis 2 months after they get the treatment and the 12 months after completion of the treatment. So you can basically see here that not only that the global gene expression of the active tuberculosis can be distinctively identified against the latently infection or normal people, but also the blood gene expression level can be used in terms of to follow up the treatment success. So most highly differentiated genes here are not just random genes. They are most of them belongs to the interferon responsive family that is in concordant with the biological significance of the interferon pathway in the immune response against tuberculosis, suggesting that the biomarkers that were found here are tied into the actual immune function and not the hallucination of the large data. In the second paper from the Kafaru groups from Imperial College London, addressed the difficulty in diagnosing tuberculosis in HIV-positive patients due to the T-cell depletion. Can blood transcription biomarkers overcome this problem? So they used just 27 transcript signatures to achieve 95% sensitivity and 90% specificity in South African and Malawi TB cohort, and also using a 44 transcript signatures to distinguish TB from similar other diseases with high sensitivity and high specificity. And in terms of robustness, again, in HIV status, diagnostic accuracy remained effective regardless of the patient HIV status. So suggesting that we may use this biomarker to differentiate active TB even in the challenging case like PL-HIV where cellular immunity are not at the normal state. So in the third paper, so a previous study identified a set of genes that comprise about 20 up to hundreds of gene expression to be measured at the same time in order to distinguish between the tuberculosis, active tuberculosis cases against latently infection or the normal people. But these strategies are not feasible to become an actual diagnostic assay since it requires a sophisticated technology and the cost to do it is so high and it's not fit for becoming a POCT method. To overcome this, the group from Max Planck investigated how minimal the list of the gene can be that can still harness the discriminatory power enough to identify the active tuberculosis. So they collect samples from the discovery cohort in India and train the data and then build the data using a random forest here. And also the conditional inference, so they can reduce the number of genes from starting with 15 genes and down to only just 4 genes, and validate it in the African cohort and giving out the 88% sensitivity and 75% specificity. So bottom line from this paper is that we can use just only 4 genes as a triage test for TB that is very possible for developing a lab on a chip or even used with existing real-time PCR machine. And it is also could be suitable for a resource-limited facility. So the next paper is in 2016, where the Stanford group published a study that identified 16 gene signatures using RNA sequencing or transcriptomic sequencing to detect the TB risk up to 2 years before the clinical onsets. So in this paper, overall sensitivity within 12 months of diagnosis before the actual clinical onset is 66%, and the sensitivity is increasing if we trying to predict a TB progression within 6 months. So this is suggests that it's not only that using this gene expression biomarker or blood-based transcriptional biomarkers to separate it or identify a tuberculosis patient, but it can be used to even tell the risk of developing tuberculosis or the TB reactivation before the clinical onset. And the last And the last paper here that I'd like to show is from Suleiman in 2018. They reported that they reported about the biggest and the longitudinal cohort of validation of this blood gene expression signature. So they used just 4 genes called RIS4 signature, and it's shown the universal validity outperforming a site-specific model. So they validated these across Africa, in South Africa, in Ethiopia, and in Gambia, and still these 4 gene blood signatures can be used. And it revealed, and it's not just that it has high sensitivity and high specificity, but the RIS4 signatures reveal the biological shift, such as B cell reduction and lung healing occurring during the disease progression. So this simplified 4-gene format is optimized for rapid, low-cost PCR testing and enable it to use in the POCT settings. So from moving from outside Thailand and coming back to Thailand. So I and my team at Department of Medical Sciences, Medical Life Science Institute has also tried it on our tuberculosis patient cohorts in the northern Thailand. So we actually, in this paper describing our work, we actually use, we first analyzed publicly available microarray data from the Berry paper in 2010, and then we identified a list of genes, about 20 genes, to be tested within our Thailand cohort. And then we narrow it down using differential, a linear differential method to come up with the minimum list of genes that we can separate it between the active tuberculosis, healthy control, and a previous TB. And then we come up with a risk score called TB6 score that comprises of 7 genes: FCGR1A, FCGR1B1, MAFB, ApoL1, STAT1, Castrin, and FCGR1B2. So using these 7 genes, we can achieve a sensitivity of 82.5% and specificity of 100%. So none of a previous TB that is already treated or the healthy control has a risk score of over the cutoff. And also, we found out that the TB6 score that we developed could have potential value for diagnosis and treatment and treatment monitoring of tuberculosis. For examples like here, we show that a 2 time point at the diagnosis and after the treatment completion. And we found out that 2 cases that failed the treatment after 6 months of tuberculosis treatment are still having our TB6 score of higher than the cutoff value, while the rest that already completed treatment and the treatment is successful, they shows a normal level of the risk score. Again, we went on and trying to test this new method in smear-negative patients. So this is just to think that we challenge ourselves in a harder situation. For example, like here we collect all the tuberculosis suspected case of tuberculosis, no matter they have a smear positive or smear negative. For patients that have smear negative, we follow them up. We collect the blood samples from the diagnosis, and 2 months after diagnosis, 2 weeks after diagnosis, 2 months after diagnosis, and 6 months after they finish the 6-month course of tuberculosis. And then we explore the outcome of that against our developed TB risk score, which are anyone who has culture positive will be considered as a TB patients, and people who has culture negative will be assessed to 2 states. First is clinical TB, because these identified by their outcome after they complete the treatment. After treatment completion, if the result that assessed by the clinician say that they have cure of the disease, then we put them into a clinical TB. But some cases cannot, after they start the tuberculosis treatment, then the clinician found out that at 2 months after along the tuberculosis treatment courses, it turns out that they have other diseases, for example, like CA lung, bronchitis, and COPD. So we put them in a change diagnosis category, which are not TB. And then we compare the levels of expression of these 7 genes, and you can see here the first one is a change diagnosis, then the smear negative or clinical TB, and then the last group is the sputum negative but culture positive. So you can see here that the levels of expression of this gene is increasing as the state of the patient is approaching tuberculosis state, or active tuberculosis. And all of this has p-value of 0.01 when comparing the gene expression levels between these 3 groups. And then we calculated for the risk scores. It turns out that all of the change diagnosis case, none of that has a risk score that is above our cutoff, while the smear negative, culture positive cases, majority of them has the risk TB risk score of over the cutoff. And we calculated for sensitivity, we have 82.6% sensitivity and 100% specificity with an AUC of 0.951. Okay. However, it seems that using the blood-based biomarker has very promising tools to be a new or tools to be adding up for use of identifying the active tuberculosis status. However, the WHO has established a rigorous performance benchmarks to guide the development of next generation TB diagnosis called WHO Target Product Profile, TPP. And in any newly developed assay that to be used as a triage test or rule out test, it has to be designed to have a high sensitivity to become as a screening tool and to confidently rule out tuberculosis in symptomatic individuals, and thereby optimizing the resource allocation by reducing the unnecessary confirmatory testing. So if you wanted to develop a test and as a screening or triage test, the minimum performance criteria that the sensitivity of test must be over 90% and the specificity must be over 70%. But for the diagnostic test or confirmatory test, the sensitivity could be a little bit less, which is 65%, but the specificity must achieve 98%. This is because this kind of test is intended to definitively rule in the tuberculosis therapeutic initiation, and it requires a much higher analytical specificity. So out of this paper from Mulenga in 2020 in PLOS ONE, they performed a meta-analysis study and evaluates 24 distinct host blood mRNA signatures that were published and demonstrated the significant potential with 17 out of 24 of the evaluated signature meet at least one TPP minimum performance criteria. But again, in head-to-head comparison with clinically relevant cohorts, for example, like with the cohorts of active tuberculosis against other diseases, only 7 signatures successfully meet the criteria of the triage test. However, there is no existing transcriptional signatures that has achieved the 98% specificity up here, 98% specificity, and that is required for a standalone confirmatory diagnosis in these diverse cohorts. So visualizing these scatter plots here highlights the lead candidates, such as the RIS-16 from SAC in 2016, or the Sweeney 3, which occupies a triage TPP zone but falls short for the high specificity diagnostic zone. So, in summary, for the tuberculosis diagnosis, these new blood-based gene expression test is a promising and it is a breakthrough. It shows accuracy such as high sensitivity and high specificity again, but it's still questionable in terms of when it comes to the accuracy and precision of the test is enough to be used in a wider setting and to be used as a triage or even confirmatory testing. And even though it is a powerful tool to rule out, but again, it still needs to be validated in a larger population studies and also in a real-world clinical studies. And again, a concerted effort between academia, research institute, and industry is required to bridge the gap and deploy these molecular tools in health facility settings. Okay. Thank you.
Thank you, Dr. Nusara, for a very nice talk. So do we have questions from the floor? Any question?
Would you like to comment on some of the functions of those genes that you've identified with the?
So for my signatures,
Yeah.
so mostly are Fc gamma receptors. So it's innate immunity genes functions. The STAT one is in the T cell functions. So it's transcriptional regulator for one of the T cells, one subset of the T cells, and also APOL1 is kind of like I think it ties to its increase in some of the it's a type of parasitic infections. So these are mostly, MAFB is another transcriptional regulator in the innate immunity pathway. So mostly these gene signatures that we found are in the innate immunity functions. So for me, I think like it's better way because it can overcome the use in the trouble in using it with the HIV status. For example, like if we talk about eager test, especially, it could be difficult in HIV positive, especially naive HIV positive cases, because the T cell is so depleted, and it can like trouble the results from those kind of tests. But these can definitely be used with the HIV, and we actually have a data on the HIV positive cases also that these 7 genes can be used.
And you might not have looked at this, but if you looked at a bunch of people with latent TB and this is active TB, is there any evidence that you could use these to identify the difference between latent and active TB?
Yes, definitely. Actually, it's not in my study, but for example, like in other studies, like these are a bigger panel of 16 genes, and it's easily can differentiate the status of the progressor against the non-progressor. Yeah. So meaning that we can detect the change in global gene expression of these 16 genes before the clinical onset of the disease. Actually, we are trying to validate our tools in this scenario also. But the problem is that to collect a lot of people, like we need a people that is before the clinical onset, so that is quite challenging.
So I have one question that what is the next step for these tests? Have you planned or developed or further or? Yeah.
So currently, this test is already offered as a service from the Department of Medical Sciences here, and I show the logo. So now it is we already service this, and some of the samples that were sent to us are one of that is a cluster of infection in monastery, like monastery, one monastery. So it turns out that one of the monk there happened to develop a tuberculosis, and then so with so the what's it called, FETP team. So they collected the FETP สืบสวนโรค what's it called สอบสวนโรค the how to say epidemiology team. So they go down to that monastery and then they collected all of the contact cases and then send it to Department of Medical Sciences for us to test whether I think they sent 7 cases and it turns out that 3 cases has the risk score of over the cutoff, and we follow up, and I think like these 3 cases they were treated as a tuberculosis patients to prevent the actual diseases.
You said you're now offering this as a test. So could you just tell us how you do that? Is that off the arrays that you've used before, or are they individual PCRs, or what approach do you use to actually convert this sort of research into a deliverable?
Okay, so in this paper, we actually use a real-time PCR because back at that time, it's so it's about like 10 years ago, to do a gene expression microarray here in Thailand is a little bit expensive.
Yeah.
So we use, that's why we use publicly available data, and then we kind of like funnel down, so shorten down the list so that to select the highly discriminatory power, the genes with highly discriminatory power. And then we test those panels, about 20 genes, using a real-time PCR. But a real-time PCR can be challenging when it comes to like measuring actual level of gene expression. So when we trying to open the service, we switch from real-time PCR into using a droplet digital PCR so that we can do absolute quantitation on those gene expression. So simply we just collect the blood, but the blood has to be stored in it's a specific tube that to store the RNA properly in room temperature even, and then it's just shipped to us at Department of Medical Sciences, and they will extract the RNA, and then they will measure the level of gene expression of these 7 genes using a droplet digital PCR.
And how fast does the transport have to happen? How long can it sit around for?
Actually, if it's in room temperature, it has to be shipped within 2 or 3 days. But if it's in like 4 to 8 degrees, it can be stored up to like 2 weeks. And then at -20, it could be up to like 2 months. The problem is that because we use like such a sophisticated machine, droplet digital PCR is not widely available, so that's why it's kind of like hard to transfer this assay to, for example, like hospital in other provinces other than Bangkok. So now it's quite centralized. Only Bangkok that has one of the centers in the Bangkok, and then another one at, I think, in Satproedprai, or it's somewhere else, another one. Yeah.
So then the transportation conditions becomes really important in terms of reproducibility. Yeah.
Well, thank you very much, Dr. Nusara. So